Hi, so I want to talk about this paper called Application of Machine Learning in Drug Discovery and Development. It was published in 2019 in Nature Reviews Drug Discovery. Okay, introduction. Biological systems nowadays generate an unprecedented amount of data, both an increase in the size of the data and also type of data. For example, we have high-dimensional omics, assays, images, text, biometrics, wearables, etc. And this data is from these high throughput approaches is both a challenge and opportunity to the pharma industry. And machine learning is providing tools to help utilize this data to help improve the drug discovery and decision making process. Uh, Drug development is famous for having a very low success rate. From phase 1 clinical trial to approval, it's only around 6.2%. And many of these trials are very, very expensive. So the use of machine learning is driven by a business need to lower attrition and cost. Um, and machine learning have been applied to all stages of the drug development process. Here, we're going to separate into four steps. First is target identification and validation. Second is compound screening and lead discovery. Third is preclinical development. Finally, we have clinical development. And I'm going to talk about each one of these parts in this talk in detail. So first is target identification and validation. As a summary, some of the tasks here that machine learning has been successful at is target identification and prioritization based on gene disease associations, target druggability and predictions, and identification of alternative targets like splice variants. So what is target identification and validation? So the preeminent approach in drug discovery is to develop drugs that alter disease states by modulating the activity of a molecular target, like a gene or RNA. And a start of drug development oftentimes requires identifying a target with a plausible therapeutic hypothesis. By that I mean that modulation of this target will result in modulation of the disease state. And selecting this target is based on the currently available evidence is called target identification and prioritization. And after that's done, the next step, we do target validation, where the role of the target in disease is validated using in vivo or ex vivo models. Ultimately, we need to validate the target using clinical trial data. However, early target validation is crucial to focusing our efforts on potentially more successful projects. There's a wide range of techniques for target identification and validation. And machine learning is being increasingly used in these data-driven experiments, and it's being applied to multi-dimensional data sets such as genetic information, transcriptome profiles, proteome, metabolome, and imaging data, etc., etc. The first step in target identification is establishing a causal relationship between a target and disease, and by that I mean that modulation of the target affects the disease from either naturally occurring or through experimental interventions. Machine learning can make predictions about the potential causality, driven, for instance, by properties of known drug targets. Next, I'm going to go more in details into a couple examples where machine learning has been applied to make these predictions. So here, one example is Casa et al. 2010. They built a decision tree classifier, to predict genes associated with morbidity, and also druggability. And they use the network topology of protein-protein, metabolic, and transcriptional interactions, and also tissue expression and subcellular localization. And in the work by Jian et al. 2014, they built an SVM using genomic data to classify whether proteins is a drug target or not drug target for breast, pancreatic, and ovarian cancers. The key features that they discovered were gene essentiality, mRNA expression, DNA copy number, mutation occurrence, and protein-protein interaction network topology. Their technique identified 122 global cancer targets, and more than half, 69 of these, overlapped with known cancer targets. In addition, for breast cancer, they identified 266 targets. For pancreatic, they identified 462 targets. And for ovarian, they identified 355 targets. And two of these predicted targets were validated in cell culture to have strong anti-proliferative effects. In the work by Amen et al. 2018, they built a model using transcriptional factor binding sites and transcriptome profiling to characterize transcriptional changes underlying Huntington's disease. The model consisted of 718 transcription factors and genome-wide target genes in mouse striatum. They use a regression model with lasso regularization. 13 of the 48 identified transcription factor modules were observed differentially expressed in striatal tissue in actual Huntington's disease. And in Momoshina et al. 2018, they compared gene expression in young and old muscle tissue to identify tissue-specific anti-aging therapies. They built an SVM with linear kernel and deep feature selection. This was shown as the best model by them. So in all these examples, the machine learning model is generating predictions of targets likely to be druggable or can modulate a disease. However, this requires further validation is necessary to generate a therapeutic hypothesis. Another example is NLP applied to biomedical literature. So literature is the primary source of knowledge on target disease relations, and NLP can automatically extract information from unstructured text to identify, for example, drug-disease relations, gene-disease relations, target-drug relations. Some of the example tools that were mentioned are BFree and DIXT.